Personalized nutrition offers a promising but still evolving approach to mitigating the profound metabolic and behavioral harms caused by ultra-processed foods. These foods, highly refined, calorie-dense, and engineered for hyperpalatable appeal, disrupt appetite regulation, promote insulin resistance, drive inflammation, and alter gut microbiota, creating a cascade of health consequences that vary in severity from one individual to another. Recognizing that genetic makeup, microbiome composition, and metabolic phenotype influence how people respond to these dietary stressors opens the door to tailored interventions that could help prevent overeating, reduce chronic disease risk, and counteract some of the cumulative damage of processed diets. However, this approach is not without caveats, and the warning is that relying solely on individualized strategies cannot fully offset the systemic, environmental, and age-related risks associated with habitual ultra-processed food consumption. Genetics play a critical role in determining susceptibility to the effects of ultra-processed foods. Variants in genes involved in appetite regulation, insulin signaling, lipid metabolism, and reward pathways influence how individuals experience hunger, fullness, and food cravings. For example, polymorphisms in the FTO gene have been linked to increased preference for calorie-dense foods and reduced satiety, making carriers more prone to overconsumption of processed meals. Other genetic variants affect taste perception, satiety hormone sensitivity, leptin, ghrelin, and dopamine receptor function, altering the neural reward response to sugar, fat, and salt. By identifying these genetic predispositions, personalized nutrition strategies can guide individuals toward food patterns that reduce reliance on ultra-processed foods, optimize satiety, and mitigate metabolic disruption. Without this insight, those genetically prone to overeating may unknowingly fall into compulsive patterns that accelerate obesity, insulin resistance, and chronic inflammation. The gut microbiome represents another critical determinant of how ultra-processed foods affect health. Processed diets disrupt microbial diversity, reduce populations of beneficial bacteria, and increase pro-inflammatory species. This dysbiosis amplifies metabolic dysfunction, weakens immune resilience, and can exacerbate cravings for hyperpalatable foods through gut-brain signaling. Individual variations in microbiome composition mean that some people experience sharper glucose spikes, higher postprandial inflammation, or more pronounced appetite dysregulation in response to the same processed meal. Personalized strategies that assess microbiome profiles can recommend targeted dietary adjustments, such as increasing fiber, resistant starch, fermented foods, and polyphenol-rich plants to restore microbial balance, improve metabolic responses, and reduce cravings for ultra-processed foods. Yet, the warning is that microbiome interventions require consistency, long-term adherence, and complementary lifestyle changes. Short-term adjustments alone are unlikely to overcome the entrenched effects of decades of processed food exposure. Metabolic phenotypes further refine individualized approaches. Some adults display rapid postprandial glucose responses or heightened insulin secretion when consuming ultra-processed carbohydrates, while others exhibit more muted but chronic inflammatory responses to high-fat, refined foods. Variations in lipid metabolism, basal metabolic rate, and hormonal signaling influence the degree to which processed foods promote weight gain, visceral fat accumulation, or metabolic syndrome. Personalized nutrition that accounts for these metabolic differences through meal timing, macronutrient composition, and portion control can attenuate some of the physiological strain caused by ultra-processed diets. However, the warning is clear. Even when tailored, these interventions cannot fully neutralize the systemic risks posed by frequent exposure to highly processed, energy-dense foods, particularly in populations with long-standing dietary patterns or existing metabolic impairment. The potential benefits of personalized nutrition are significant: improved satiety, reduced overeating, better blood sugar control, lower inflammation, and optimized gut microbiome health. But these approaches must be integrated with broader behavioral, environmental, and policy measures. Without structural changes, such as reducing availability of ultra-processed foods, improving access to whole foods, and addressing social determinants of health, the effectiveness of individualized interventions is limited. Furthermore, reliance on genetic, microbiome, or metabolic profiling alone can create a false sense of security, leading individuals to underestimate the inherent risks of ultra-processed foods or delay necessary behavioral changes. The warning is unmistakable. While personalized nutrition can help mitigate some of the harmful effects of ultra-processed foods, it is not a panacea. Genetics, microbiome composition, and metabolic phenotype determine susceptibility, but these factors interact with environmental, cultural, and behavioral drivers of overconsumption. Individuals who consume processed foods habitually, even with tailored strategies, remain at risk of insulin resistance, obesity, chronic inflammation, cardiovascular disease, and accelerated aging. True protection requires a combination of personalized guidance, long-term adherence, and systemic efforts to reduce exposure, promote minimally processed diets, and address the underlying social and environmental forces that drive reliance on ultra-processed foods. Without comprehensive action, the cumulative metabolic and neurological harm caused by these foods will continue to escalate, even among those receiving the most sophisticated individualized interventions.